So 6.3 is about eukaryotic cells and their nuclei and their ribosomes. So remember that the things that are unique about a eukaryotic cell are that it has a membrane-bound nucleus. So that means it has a thing in the middle of the cell which is encapsulated or covered, surrounded by a membrane, a double membrane, which is just like the cell membrane, same material is making it up, and that's where all of the genetic material, all of the DNA is stored. And this is different than a prokaryotic cell, which does not have a nucleus. So eukaryotic cells are unique in that. And we also have ribosomes, which prokaryotic cells do also, but we're going to talk mostly about eukaryotic cells now. We have ribosomes, which make our DNA into proteins. So remember, proteins do everything. You should know that by now. Proteins do everything, and so we make proteins on the ribosomes. Okay, so the nucleus says information central. It's exactly what it is. This is where all of the information for the cell is stored in the form of DNA. And so it contains, it says most, and we'll talk about where the rest of them are, but all of the important things for the whole cell are inside of that nucleus. And it says usually the most conspicuous. So if you're looking at a picture of a cell, it's going to be, let's flip to this slide, the one right before this chapter. You're always going to find that you can see the nucleus, okay? This whole big purple thing is the nucleus in these duckweed cells. That's a plant. This is the nucleus right there. It's the biggest, most identifiable organelle inside of a cell. And it's held in by the nuclear envelope, like I just said. And so it separates everything that's inside the nucleus from the cytoplasm, which is outside of the nucleus. And this is a double membrane. So this gets kind of confusing because it's made of a lipid bilayer, so that's what we were talking about before, with the hydrophobic tails on the inside and the hydrophilic heads on the outside, but there's actually two of those. So there's two layers right here, that's one membrane, and then there's a second membrane right next to it. So there's actually two membranes. That's what we mean by double membrane. It doesn't mean bilayer, it means two actual membranes, which you can see. There's an inner membrane and an outer membrane. So it's pointing right here to the inner membrane and then to the outer membrane. And each of these is a lipid bilayer. Phospholipid tails on the inside, which are hydrophobic, hydrophilic heads on the outside. These are the phospholipids that we talked about. Two hydrocarbon fatty acid tails, glycerol in the middle, phosphate group for a head. Okay, so the nuclear membrane surrounds the entire nucleus. And there's these pores in the nuclear membrane, which are really important. So there's a nuclear pore, there's a nuclear pore. These are connections where the two bilayers actually connect to each other. So let's say I have one layer right here and one layer right here. There's going to be a hole, and the two bilayers are going to be connected to each other. I don't know how to demonstrate that. But that allows RNA to get out, because if all the information is stored inside there and it's behind locked doors, what good is that going to be for anyone who needs that information, right? So the nuclear pores allow specific information in and out. And they are, this is the nuclear envelope. Here's one of the pores. It's called a pore complex. It's made by a bunch of different proteins and such. So there's some pore complexes you can see. It's not just one smooth thing. It's made up of several different proteins. And then these pores regulate, like I said, the entry and exit of molecules from the nucleus. So remember, we're going to need to get that RNA out, because it's like if you had a library with locked doors and all the important information was inside those doors. What good is that going to do you? Absolutely nothing. So the shape of the nucleus is upheld by something called the nuclear lamina. And so if we didn't have the nuclear lamina, it wouldn't be so easy to maintain the shape of the nucleus, and that's really important to protect what's inside. So the nuclear lamina is a bunch of proteins on the inside of the double membrane, which just support it and keep it shaped. And inside of the nuclei, the DNA is organized in discrete units, meaning several distinct different chromosomes. Each chromosome is composed of one really, really, really long DNA molecule. So remember, DNA is a nucleic acid, so it's made up of a bunch of nucleotides stacked on top of each other. Tons and tons and tons of nucleotides bonded together by phosphodiester bonds into one long one long nucleotide, which would be DNA. And it's organized by protein. So imagine you have this ridiculously long piece of string, which is all these nucleotides stuck together, and you need it to fit into a really small spot. And there's a bunch of them. There's a bunch of chromosomes, which are those long pieces of string. You shove them all together, shove them into a tiny spot, and then you need to pick out one. It's going to be very challenging. You need some way to organize it, otherwise they're all going to get tangled. And so the proteins help prevent the DNA from getting tangled. So there's a lot of proteins interacting with the DNA inside of the nucleus. And all of this together is called chromatin. So there's a lot of words that sound similar here. Chromosome, chromatin. Chromatin is the DNA and the protein together. And then when it's time for the cell to divide, the big long pieces of string, which are chromosomes, they condense into the X's that we know as chromosomes, that you see and recognize as chromosomes. So chromatin is when it looks kind of like a big bowl of soup inside of the nucleus, and chromatin is when you can actually see the X's. And it says the nucleolus is located within the nucleus and is the site of ribosomal RNA synthesis. So the nucleolus is the dark spot inside of the nucleus. Where's that picture? Sorry for jumping around all over the place. Here's the nucleolus. It's the dark spot inside of there. And that's where we make our RNA, which is part of a ribosome. And so you need that part of a ribosome to make proteins in the future. And so the rRNA is made inside the nucleolus. So that's very important. And then we get to ribosomes. Ribosomes are actually where the proteins are made. And they are made of two subunits that are made of protein and RNA. And these are key. We need both of them to do their job. And then there's two locations we might find ribosomes in. They might be floating around in the whole entire cell, because if you're going to need a particular protein in one particular part of the cell, it makes sense to make that protein there. So there would be ribosomes there to make proteins for that part of the cell. Or they might be bound onto another organelle that we're going to talk about later, called the endoplasmic reticulum. So there are bound ribosomes, which are stuck on the endoplasmic reticulum or on the nuclear envelope, or there's free ribosomes, which are just around in the cytoplasm of the cell. And so you can see here from this picture, these are the free ribosomes just floating around inside the cytoplasm, inside the cytosol. And then here's the endoplasmic reticulum. You can see that there's ribosomes just actually stuck on the endoplasmic reticulum. And this is what our ribosomes look like. You can see a small subunit and a large subunit. Oh. That's the end. We're going to talk more about ribosomes in Chapter 17. That's all for 6.3.